Oh, in a winner. And then just to remind everybody of some of the etiquette that we have for these WebEx meetings. If you can type any comments that you have in the chat, that would be helpful towards the end of any questions you have towards the end of the seminar. Please turn your mic off when you're not speaking, and yeah, and pay attention. So anyway, some things about Sonia. Sonia, as you will hear in a little while, is Portuguese. She graduated in agronomy and crop breeding in an I'm not even going to say what the organization is anymore. The Institute of Technology and Chemistry and Biology is the English translation, which is near Estoril in Portugal. Again, in her PhD, she focused on breeding Portuguese heritage rice in collaboration with the International Rice Research Institute, IRRI, in the Philippines. Sonia then did a postdoc in the same institution in collaboration again with IRRI, targeting rice genomics and allele mining for salinity stress. And after that, in 2013, she became a research scientist at the King Abdullah University of Science and Technology in Kaus in Saudi Arabia. I think it was where we first met, actually, when you were working there, Sonia. And there she worked with Mark Tester and a whole range of different people, contributing to many projects involving the genetic dissection of the mechanisms of salinity tolerance. And in 2018, Sonia was not lucky at all, fortunate maybe, to get a position in University College Dublin, where she's now an assistant professor and PI of the Crop Stress Interaction Group. We've had good contact with Sonia ever since she arrived in Dublin, and our group is now focusing on crop physiology, abiotic stress and adaptation in barley using both phenomics in controlled and field conditions. So Sonia, it's a pleasure to have you here with us. We're all looking forward to the presentation you're about to give us. So over to you.
Thanks, Robbie. First of all, thanks for the invitation. I hope you can see, all of you see my my presentation, yeah?
We can.
Okay, good. So thanks again for the invitation. It's lovely to be here and talking a little bit about barley phenotyping. So first of all, what is plant phenotyping? So if you think that a plant's genotype is all of its genes, then a plant phenotype is how a plant will look and perform. So plant phenotype is a combination of the genotype and the environment that the plant is growing in, which means that the plant with the same genotype will have different phenotypes. For example, if we have RGT planet being grown in County Kildare, we'll have a different yield and a different phenotype is if we would be growing RGT planet in Wexford, or of course, if we would grow RGT planet in Spain or Germany. The phenotype would be different, the yield will be different, the disease pressure, etc. So phenotyping is analyzing a plant's phenotype. Now, today we are going to talk a little bit more about phenomics or high throughput phenotyping. So high throughput phenotyping is a way to speed up phenotyping where we use high-tech imaging equipment, different types of camera sensors, and we also rely upon computing power and computer science as well. So this is just an image of just showing how you have the genotype and the interaction with the environment will produce a phenotype, and we can assess how a plant performs and how the plant is resilient or not in terms of its response to the environment using plant phenotyping. So when we think about plant phenotyping, normally we think in the classical methodology. The methodology that brought us up to where we stand now. So we have been phenotyping plants ever since we started to domesticate plants 10,000 years ago. So we used our eyes to phenotype plants. So we selected plants that had a low seed shattering. Later on, we start selecting plants based on yield. We looked into plants and looked into disease resistance, so visual symptoms of the disease. So we have been using classical phenotyping ever since. Now, normally classical phenotyping is destructive. So we harvest a plant, we collect the yield. So it's a destructive analysis. And even if we are looking into visual scoring, it's just a snapshot. It's on that moment, on that day, what was the response of the plant to the stress. Now, when we are talking about high throughput phenotyping, we are talking about imaging sensors, different type of cameras that I will just explain now. And high throughput phenotyping is non-destructive, meaning that we can follow exactly the same plant throughout time. So in the end, we have time series, we have longitudinal data, and this causes a big data problem as well. So when we are talking about classical phenotyping, we can talk about shoot. Everybody has done that in the past. Shoot and root mass. We can also use handheld devices. In this case, we have the SPAD over here, which is a proxy to chlorophyll content of the plant. We can have fruit and yield related traits. Over here is a big harvest that we had in Saudi Arabia. Over here is my PhD student, Patrick, harvesting by hand barley. And of course, visual scoring. This is still coming from my postdoc, where we would phenotype in a visual way if the plant was tolerant or susceptible to the stress by giving a number. And as you can see, it's time consuming, is laborious to do all these classical methodologies. So when we talk about image-based phenotyping, I decided to divide in two different ways because that's how I see it. So in one hand, we can call just plant imaging. That can be used in a smaller scale for physiological studies. For example, we might have just two contrasting barley, or we can have the same barley accession under control conditions and under stressed conditions. This is the raw image. This is the treated image. We'll go there in a minute. And this can be used and very powerful for physiological studies. And we are talking about a smaller scale, small number of genotypes, okay? Now, when we talk about high throughput phenotyping, we are going into a larger scale. So we are talking about hectares of farmland or big facilities that can cope with 300, 400 plants at a time. And these large-scale phenotyping can be used for breeding and genetic studies. So if you're doing breeding, you cannot have a small number, you have to go on a larger scale. So what are the types of imaging sensors? So the most common imaging sensor is RGB, which stands for red, green, blue. It's the same type of camera that everyone has in our cameras, in our cell phones, and goes into the visible range. We can also have other imaging sensors that have, apart from the visible range, they also have near infrared, and they can be called multispectral cameras. We have shortwave infrared that goes up to 2,500 nanometers. This goes into the realm of what we call hyperspectral imaging. We can also use thermal infrared imaging, and all of these methods are passive. So we take that image and the plant is not responding in real time to us. Now, when we talk about chlorophyll fluorescence, what we do is that we image the plant and we also have different wavelengths. So we expose the plants to different wavelengths. And the plant will respond to those different wavelengths with fluorescing. And then we can record an active response of the plant to different wavelengths. We can also use 3D technologies. So that's when we are using LiDAR or scanning with lasers. So these are active responses, or CT scanners as well. So these are just a couple of examples of what we use more in phenotyping. So this is the type of image you obtain with Rgb image. Again, this is the raw image. This is the treated or segmented image. When we talk about chlorophyll fluorescence, as I explained, we expose the plants to different wavelengths and we can assess photosynthetic responses. So the plant emits fluorescence and we know the F0 stage, we know the FM, the maximum fluorescence, the minimum fluorescence, and the quantum yield. So it's an active response. We can also have thermal infrared imaging that is very used in terms of transpiration, water loss, because you can see the differences in terms of the temperature of the plant and different lines or mutants, transgenic plants, etc., will have different temperatures. And then finally, hyperspectral imaging. So I'm just putting the most commonly used imaging sensors. There are many others, especially if we go to roots. But in this case, for the purpose of this talk, we are just focusing on shoots. So on hyperspectral imaging works in 3 dimensions and not in 2 dimensions. So you have the plant space, so you have the X and Y dimension of the plant, and then you have what is called the hypercube. So each one of these layers is one wavelength that we are capturing. So RGB, you have 3 channels, or red, green, blue. Hyperspectral, you can have up to 240 channels. Each one of them will give you one layer of data. So that's why you can see why I'm leading to the big data problem. So a PhD student in my lab is doing a systematic literature review, and what Jason started was to try to see the trends in plant phenotyping. So you can see that most of the methods are using hyperspectral and RGB method imaging sensors, and you can see the trend line in terms of years, how it has been increased. Now, I have to mention that this is not only imaging, this is imaging using machine learning, using computer science to analyze the data. So you can see that in very early on, you basically didn't have nothing. So these phenomics and the use of machine learning is quite a recent thing that has been happening in plant sciences. So the general workflow of imaging data is that you take your photo, you have the raw data, you pre-process. So you have to crop the image. Sometimes you have a fish eye lens that you have to correct. For example, so that's the pre-processing. You then do the segmentation, which is when you extract the plant pixels from the background. So you want to know only the plant pixels and not the background. You have the feature segmentation, where you try to get a trait list. You have to do the outlier and quality QC. So for instance, when you're talking about high throughput phenotyping facility and you have 400 plants and you look at the data and you have an outlier, you have to probably go back to that image and realize that that outlier was just a plant that broke or that didn't germinate properly, and then you have a runt. And so that plant has to be removed from the analysis. You then estimate your traits. So for instance, the convex hull, which is how the plant shape will look in the image. You have different types of vegetation indices, like the normalized differentiation vegetation index, etc. You then have to, and this is where computer science really has a big, big role. You have to mine the data, because we are talking about gigabytes and terabytes of data, and you have to manage the data in an accessible and open way that can be reproducible. So we are talking about phenotyping in controlled conditions and in field conditions, and non-destructive quantitative measurements over time. So this is quantitative. When we are talking about visual scoring, I can score in one way and Robbie can score in a different way. So then we have a user difference when the plant was the same. So this is actually very precise and quantitative. And in my project, what we are studying in my lab is we are focusing on waterlogging. Why? Because barley is particularly sensitive to waterlogging, and we think about Ireland, we always think about the seasons of Ireland and constant rain. So the waterlogging is really impacting barley production. So this is a photo from 2019, and you can see that this is spring barley early on a season. So this area, this patch of the field, the plants at an early seedling stage were exposed to waterlogging, and later on, all of that area, you didn't have anything growing there. So it is a problem, and it is an increasing problem. So this map over here shows the changes, how rain patterns are being changed with climate. And what you have is that you have Ireland over here. It's becoming more blue. So we have drought, of course, and we have drought in the season. So, but what is happening is an unpredictability of the weather. In one season, you might have a severe drought. In another season, you might have, on the following year, you might have a waterlogging. So we have to try to understand how can we improve the plant's performance, and how can we assess how the different genotypes behave in response to these difference in rain patterns. So climate change is affecting barley production. And how does waterlogging impacts the plants? So when we have control conditions, you have oxygen in the soil, you have favorable redox conditions, you have a balanced nutrient uptake. However, when we have waterlogging conditions, meaning that only the root area of the plant is exposed to the water, not the whole plant. That's called submergence. So it's just this lower part, just a small patch of water, layer of water in the surface. You have shallow roots. You have an imbalanced nutrient uptake, so the plant goes into nitrogen deficiency. Other nutrients can become toxic, for instance, iron. You have the classical response of plants of forming a parenchyma, meaning that the roots will have to perform respiration. There's an energy crisis in the plant. There is no respiration because there is no oxygen. So this energy crisis can lead to fermentation processes. So one of the most classical responses is the parenchyma, which is this shallow pipes that will lead the oxygen into the roots so that the roots can perform respiration. And of course, in the end, what happens is that you have reduced height, biomass, tillering, and yield is reduced. So the project that I'm talking a little bit today is to elucidate barley stress adaptation in the context of climate change using these disruptive technologies, image-based sensors. So it is an interdisciplinary research where we are doing high throughput phenotyping under controlled conditions, under field conditions, using unmanned aerial vehicles, also known as drones, so just using it drones. And then we are using machine learning and computer science to understand the plant's response and to mine the data in this big data problem. On the other hand, we have, of course, genotyping and barley genetics and genomics. So this is where we have been collaborating with Robbie's team at the James Hutton Institute, assembling a collection of barley so that in the end we can perform association mapping. So what we are focusing on is that we are focusing on heritage barley. So we have assembled a collection of heritage barley that you see here, this photo, which is the same one as you can see it behind me, or a similar one. And our underlying hypothesis is that modern cultivars were bred under optimal conditions. So they have response to high yield, but they also require a high input. And heritage germplasm is more adapted to more sustainable practices, lower inputs, and because of that, they might have, our hypothesis is that they might have higher stress tolerance and a better resource use efficiency. And this heritage germplasm remains largely uncharacterized. So we are talking about land races and formally bred cultivars. So the first step that we did together with Robbie's team was to assemble the European Heritage Barley collection. You can see here some photos with a diversity on the spikes, and you have different colors and shapes and sizes. So this heritage collection comprises 365 accessions of 2-row spring barley, mostly from Northern European countries. All of them were genotyped with 50K iSelect SNP arrays, and we have performed population structure that suggests 4 groups in these lines. So this is still preliminary studies, and we are still analyzing the data. So you can see that most of them are coming from the Northern European countries, just because it's where we hypothesize that we'll have better waterlogging resistance traits. If we were focusing more on drought, we would for sure focus more on Southern European countries. So this is field data from 2020 where we performed classical phenotyping, and you can see how the collection is quite diverse and has a very good phenotyping diversity. However, we thought that the exhibit collection of 365 genotypes was a little bit too much for us to handle in terms of data. So we assembled a core collection by performing a power analysis. So this was again with Kelly Houston from the James Huston Institute. And so these 230 lines will, as you can see, they have genetic diversity. So all of this was done based with genetic markers, but also we also made sure that you captured the whole phenotypic range diversity. So what did we do next? We phenotyped them. So we assembled the collection and we phenotyped it. So we used the imaging system platform that we have at UCD. So the imaging system is not a high throughput system, so we can only use it in a smaller scale. So in the realm of plant imaging, and we have 3 types of sensors here. So we have the first station, we have chlorophyll fluorescence that we can assess photosynthetic traits. So we can assess quantum yield, FAM, so we can assess how is the photosynthetic apparatus of the plant responding to stress. We can also look, we also have a Rgb imaging station where we can look into growth. Remember that we can image the same plant every day. So every day we can capture the number of pixels, which we know it's a very strong correlation with fresh biomass. So we can look into growth and biomass. We can look into plant height as well, and we can also look at morphometry. So for instance, if the plant has a plant architecture droopier or more erect. And the Rgb station, we have a top camera and a side camera and a platform that can rotate 365 degrees. So we have then the hyperspectral camera. So here we can look into stress indices. So we can look into the normalized differentiation index, which is the most commonly known for stress. But we can look into others, and we can look as well to pigments such as anthocyanins and carotenoids. So this is what this is how the system looks like. You have a conveyor belt, and you have over here a laser that reads here this QR code. So in this case, we were just testing the equipment and testing the protocols. So you can see that we have a control pot and we have a waterlogged pot over here. And So all of this is in blue because the plants do not have blue pixels. So it's much easier to extract the colors and segment the image. So this is just a small video to just show how the plant will go inside. And you have here the chlorophyll fluorescence station that will emit all those different beams of actinic light that then can capture the plant's fluorescence response. So if we look at this top camera, this is what we would see. This would be a top view of the top camera. And so we started by optimizing the protocol. So whenever you start working on these big equipments and you're doing such a large experiment, you have to make sure that you're nailing the stress conditions. So we tested different protocols, different stress periods, different times should we put recovery or not. From this protocol, we just used two lines, a heritage line and a lead line, and we had 240,000 images just from a small experiment of testing a protocol. In the end, we did classical phenotyping, so we harvested and we measured the plants because we wanted to make sure that what we see in the imaging can correlate really of what we harvested in the end. So we decided on a protocol that was 14 days of stress and 7 days of recovery. And you can see here our Irish heritage line, Glasnevin number 5. And you can see RGD planet. And you can see that in terms of the shoot fresh mass of what we harvest on the end point, you can clearly see that there's a larger reduction in the RGD planet compared to the heritage line. So this was just a small experiment of just optimizing the protocol. So we then looked into different Irish sessions, and you can clearly see, so if we do that pixel count, which is a very good proxy for biomass, you can see the plants throughout the time growing, their exponential growth. In orange is the stress period, the 14 days of stress period. In green is the recovery period. And you can see in full all the lines in control conditions and all the lines in waterlogged conditions. And it's very obvious that all the plants suffered a reduction in biomass. But what's interesting, and that's the power of imaging, is that then once we start analyzing the data, we can see at which day did that change between control and waterlogging became obvious, became statistically significant. So if I would harvest the plants at 8 days and do a destructive analysis, I would have these results. But if I would do it at 21 days, I would have a different type of results. So it all depends on not having that snapshot, but having this time series. Now, from hyperspectral cameras, we also got this photochemical reflection index. That's a very good proxy for stress response, and it's related with the carotenoids and the capture of the CO2. And you can see that in just one day, you have a massive drop on the system, on the plants responding to the stress. And then with time, they start to balance back. And if you go later on the stress, the photosynthetic system is trying to cope with the stress. And if you go to the recovery period, it's basically similar. So this is a sort of a very immediate response of the plant that then the plant goes up and tries to cope with it. So, but there are challenges. There are several challenges. So the first challenge is improving the image segmentation. So I explained to you what is the image segmentation, is removing that background of the plant. And what we have is that we are having a problem on the water reflecting the light in the image. So basically, if you see the control, you have the waterlogged plant over here, and the segmentation is not capturing any pixels. So this is just because there is a reflection of the water with the lights. And so although the plant is here and you can see it, the computer is not seeing it. So it's not capturing. So we need to improve the segmentation. So this was an unexpected problem that showed up when we started looking into the data. Of course, this is a big data problem. And we have to use new analysis methods. So we have to use longitudinal data and we have to use hyperspectral data. Computer science. So the other challenges is that we have algae in the surface. So sometimes you could have algae growing up, especially in the waterlogging images, and they will contribute to extra pixels. So again, the computer is collecting those green pixels as plants when they are not. And of course, if you have more water, you can have spillage and you can have extra weight of the pots that can cause problems on the conveyor belts. And the conveyor belts, believe me, they're very, very sensitive. So the image segmentation, we have to improve it. So this is what we have been doing in our lab. So this is the system, the default system of the instrument. So that when you open the data, the morpho analyzer of our equipment, this is what the system uses. And most of the systems use this type of segmentation. So they use a color segmentation. So they choose everything that's green, it's a pixel of the plant, and everything that's blue or gray or black, it's background and it's unwanted. So in the end, you have the segmentation where you can then count the pixels and knowing exactly how many pixels do you have. Now, as I told you, we are having these challenges of the water reflection and more importantly, the hyperspectral imaging, because you have the fish islands because you don't have a real 2D image, you're imposing a 2D image. So the segmentation is being quite challenging. So normally the classical method that you have and that it's used by the equipment is that you have the raw image. So again, this is a hyperspectral image. You have this mask, this binary mask. The problem is that when we overlay the mask with the real image, you can see that it's very poor. The alignment is extremely poor. So the segmented image is far from perfect. And that's why when I showed you all of that data of that graph with all of those Irish lines, I said it was preliminary data. Because what we are using is this image, and we know that this image is not a real, it's not a real input data. It has bits and pieces missing. So this is the classical method is a color segmentation that's commonly used by most imaging equipment manufacturers, and the output is not optimal and you have a hyperspectral issue. So the binary mask, which is what we see here, is not aligning and the background is not fully removed. So in some cases, we also have some pixels being here picked up. So what we have is that we have Jason. He's a PhD student from computer science. So he picked up the imaging learning, and Jason's background is computer vision and machine learning. So he decided to pick up the image, the hyperspectral image, the raw data, and then this is when he uses the typical segmentation of the equipment from PSI. So you can see again, this zoomed in, you have some bits and pieces of the equipment being captured as pixels over here, and then you don't have overlay. So you have a lower accuracy. So what he did is that he used deep learning methods. So he created a training set of 500 images and he trained and he's now in testing those images. So, but when he uses this segmentation with machine learning, you can see that his accuracy is much higher. It goes to 98%. So you can see that the use of the machine learning and training with these so many thousands of images that we have. So this is a small subset. So this is still preliminary. It's ongoing work from Jason. Is that we can improve the segmentation using deep learning and machine learning. and it's much better than the classical segmentation, which is color-based. So now in the field, so we have 2 years of field data. So we have been characterizing the exhibit collection in the field at UCD Lions Farm. So last year we had more breeding plots. We had very small plots, and this year we had the control plots. We had much larger amount of seed, so we can go into larger plots that could be harvested with a plot combine. So in our conditions in Kildare, where is located the UCD Lions Farm, we saw that at the end of April, last year we did a pilot test. Again, we wanted to make sure about the water percolation in the soil and all of that. How many days did we need to water? We imposed the stress at early seedling stage. We collected data throughout the season, and then we harvest. So this is last year. We had to again optimize the conditions. So we tried 2 days, 5 days, and 10 days of water logging, and we had the control. And you can see the water content on the soil with the different days. And what we were able to look is that we were able to pinpoint that the best way of having our waterlogged stress would be between 2 and 5 days. 10 days was already too long, and in terms of farm management was too larger amount of water. So this year, our irrigation logistics were that we used 2 tanks of each tank with 26,000 liters, 1,700 meters of pipes, 2 water pumps, 1 electric generator, and you can see. And actually, when we were doing this, we had to delay. It's very funny, but we actually had to delay the water logging imposition. You know why? Because it was pouring rain. And because it was pouring rain, the irrigation people could not go and install the pipes and the pumps and all of that. So we had to wait a little bit longer because it was so wet for us to start wetting the data. Of course, we have all the weather data that also goes into computer science and the machine learning models. So you can see how puddled it is. So what we ended up using was 4 days of irrigation, 12 hours per day. We used a total of 250 cubic meters of water. And you can see the volumetric water concentration in the soil between control and stress from the days of waterlogging. And it's funny that you can see that actually the control plots afterwards in the end of the season, they were slightly wetter than the waterlogging plots. So if we look carefully into the first days where we did the waterlogging, you can see, and this is the soil data. So this is actually physically how much water did we have in the soil. So this is again preliminary data. It's lots of data, as you can imagine. So we have these 230 lines. And what is very clear is that you have a massive reduction in all the parameters when we compare control and waterlogging. And also even on the having date, when we were scoring, myself and my colleague, we could see that the control plots were flowering much earlier than all the waterlogging plots. So there's a clear effect of the waterlogging of just 4 days of waterlogging in all the parameters of the field. So this was classical phenotyping, and we had to phenotype with imaging systems. So we used a multispectral camera, we used an Rgb camera. So this is just a small video to show. So over here is our control plots. Of course, we used exactly the same sowing rate in both control and waterlogged. Over here, you can see the waterlogged plots. And this is just a small video. Believe me, the drones don't fly that fast. I speed up the video, otherwise we would stay here for the whole afternoon. So and you can see the logistics of the pipes over here and the generators, the tanks, etc. So we have to process the data. So the first thing that we do is to create an ortho mosaic. So what the drone does is that when it flies, it takes photos. And then all of those photos have to be sort of stitched in like a patchwork. The same way that you do it with quilts and patchwork, it's has to be done the same way. So to form this ortho mosaic is all these thousands and thousands of images that are stitched back. So our wet plot, we have a sort of an area of 1,500 square meters. The control was 1.5 actors, and this is the Rgb imaging, and this is the NDVI imaging. So this was captured with a multispectral camera, which has the RGB plus the infrared. So captures on the visible range and the infrared, the near infrared range. And because we capture that, we can have this very important index, which is called the NDVI, Normalized Differentiation Vegetation Index, that uses this formula and gives you this image. Now, this was our third flight. So this was the flight that happened 3 weeks after the end of the waterlogging. And you can see that after 3 weeks, our wet plot, even by eye, we still didn't analyze the data. This is just by eye. You can see that the color of the NDVI is different from the control plots. And you can see that these areas over here that had also a patch of land that had more rain coming in also was affected. So of course, we still didn't analyze the data. This is really fresh data. This was captured during this season, this growing season that we just harvested in the beginning of September. So this is just to show the progression of the field throughout the different flights, the control field. You can see how you can capture all of the plants growing, and you can also see the same into the waterlogging images and how the plants behave. Now, there are also several challenges. When we think about waterlogging, we cannot use ground-based phenotyping. So ground-based phenotyping in a farm context is where you have a tractor with a beam, where you have the cameras on that beam. You can also have a platform that moves, okay? Or you can have a sort of a phenomobile that could be even handmade, where you have 4 wheels and you have a certain height and you have the camera on that height and you go into a constant speed that you analyze the field. Now in waterlogging, because we have all of that muddy soil, we cannot use any vehicle. We cannot use the wheels. So we really have to phenotype using UAVs. Another thing that we are seeing is that when we are using the machine learning algorithms, irrigation equipment can become unwanted objects that can be interfering on the training of the model. The other challenge of UAVs is that they are dependent on the weather, and there are unfortunately several weeks that we couldn't fly because the wind was too strong, or was it raining too much? And we are talking about spring and summer. So in the winter time, using UAVs in Ireland is extremely challenging. And then the location of the field. Now we are very unlucky because the Lions farm is located here. Our field is actually located here, which goes into the 5K area of the Casement Aerodrome, which is a military aerodrome. So there are some times that we do not have permission to fly. And whenever you're using UAVs, you always have to have the permission to fly from the aviation authority. So that can be challenging as well. The next thing that we are talking about is cost, of course. buying a UAV is not as cheap, especially if you want one with a good camera that can enable you to really have a higher resolution in terms of pixels. It's not a cheap equipment. You have to have the technical manpower, drone flying permits, and then all of the software to analyze and high-end processing computers. So for instance, Jason, who's working on the data, and Mortaza, both of them have to have a different type of um, laptops or desktops that can cope with really large data sets. So this is not the type of thing that you can do in a normal laptop or a normal desktop, because again, we are talking about terabytes of data. So this is a big data problem that requires different expertises and technical capacities from computer scientists, etc. And the other very important thing is that, yes, we are talking about image-based technologies, but we always have to ground truth the data. And this is particularly important in the field. So when I was talking about controlled data, I was saying that in the end, we harvested the plants and we measured to get all that shoot biomass that then we could correlate with the pixels. And when we are talking about field is exactly the same. So you still have to have your harvest data. You can collect other types of traits. In this case, for instance, we are looking at the leaf area indexed, which is a cover. It's a proxy for the plot cover. How much is if the plot is well covered or not? And we have to have the weather data. So all of that is required to improve your spectra and your data analysis. So there's lots of to-do things. So we are still analyzing the drone. We just basically just build up the ortho mosaics. We have to do all the spatial corrections for the field data. We are conducting imaging experiment in control conditions in collaboration with our colleagues at in France at Université Picardie Jules Verne. We are using the control data, all of that data that I showed you, all of that is preliminary. We have to get the new segmentation pipeline and then pass over all of that data through the new segmentation pipeline so that we can have a better estimation of the traits. And of course, we are doing association analysis. So the exhibit collection, we have to validate its power for future association studies. So we have to validate this. And then, of course, we'll have to do all of this again next year. Lorries transporting water tanks and meters of pipes and all of that. So there's a lot of people to acknowledge. Not going into detail, all of my team, I presented here several things coming up from my group. The collaborators in UCD, Eleni and Kevin from the School of Computer Science and the School of Agriculture, and people from the Lions Farm, Robbie and his team at the James Hutton Institute, University Picardie, and more importantly, funding for the budget. And thank you all, and I welcome all the questions you have. We'll stop sharing now.
Sonia, thank you. That was fabulous. I should have said at the very beginning that, of course, UCD were lucky to have you. You weren't lucky to get the job in UCD, so I apologize for that at the beginning. I should also say to everybody that's listening on the call that I think, 3 years Sonia's been in UCD and we've made good links already. And I think in Dublin, she's made already a big impact. So I think it'd be really good for us to increase our level of collaboration. You're doing things in the field that we want to do in the field, and I think we could learn an awful lot. So does anybody have any questions? If you use either the chat box or put your hand up in the short term, I can't see if you put your hand up in live because nobody's got their cameras on. But if you want to put your camera on and put your hand up, then please do. Okay, Michael.
Yeah, thanks. It was very interesting, Sonia, especially since I can recall back in the late 90s, there were already some work done on that by the University of Bonn back in Germany. So we are familiar when I worked in the malting industry, when poor students had to collect data on the ground as reference data by kind of integrating the leaf surface on a piece of paper. I think
Yeah.
that's where the artificial intelligence and the machine comes in handy. Right. I was very interested in your presentation. You mentioned two-row spring barley. Are you looking into two-row winters as well? Because we are actually promoting them for climate risk mitigation with some good results here in Ireland.
Well, that's a very good question. So we focus specifically on two-row spring barley because in the case of Ireland is still the most commonly used type of lines for malting purposes. And also there was a project in collaboration between Chigask and UCD before I arrived here. So this was, I think the project finished in 2018 or early 2019. And they were looking into winter barley. So they actually look into waterlogging and winter barley in Chigask, Susan Barth, and they so because they had done already that bit on, we decided to focus on the spring barley, and also because winter barley in one hand makes more sense because you have more exposure to rain during the winter time, but there is a very important link between waterlogging and temperature. So the lower temperature, the easier it is for the plant to cope with the stress. So if you have waterlogging in spring, the effects are more drastic than if you have waterlogging in November, where the temperature is much colder. So because of that, because of all of this together, we decided to focus on spring barley. Yeah.
No, I'm just mentioning that because we are have trial and also signed off on a two-row winter malting barley variety, which we think is a good fit for the area around Athy, because we experienced actually a lot of drought stress in the area in the last years with very disappointing results, and we just think that a barley variety which is less subjected to the usual summer sprint maybe more suitable in terms of sustainability, quality, and yield. And so just ask, because it's that the water logging, that's also a function of the soil, etc. So I think the Athy areas in particular, rather over the last years, has been rather being hit by drought. Rather than
for us, it was funny because we were so, as I told you, on 2021, we literally had to delay the stress imposition because people could not put the irrigation pipes because it was pouring rain. On 2020 was the other way around. We sowed on the most severe drought in like 50 years.
Yeah.
And during the summer, we had extensive rain and storms. So scoring the heading date was very, very challenging because, again, we could not go to the field because it was rain and high winds and all of that. So we have all the weather data, and the last 2 years that we have been doing field trials, it's a little bit all over the place. We can have drought and we can have waterlogging and we can have in the beginning of the season, in the end of the season, we never know.
I can recall 2020, everybody was very happy because it was one of those years when we had very early plantings and then we were badly hit towards May, June. I can recall that. Yeah, you're right. It's the weather patterns become increasingly unpredictable. Well, 2021 here was a good crop year, I have to say. Very happy. Thank you.
Thank you, Michael. So the questions have started to come in, Sonia. I'll just ask you. The first one is from James Brosnan, and he asks, how much more susceptible to disease are water or drought-stressed crops, and can the monitoring successfully differentiate between them?
That's an excellent question. I can tell you that, unfortunately, I will have to put a new project to look at that. But preliminary data, and you can see my excitement already, preliminary data showed that waterlogged lines are more sensitive to stress. If I'm not mistaken, yes, they were more sensitive to stress.
For biotic
And
stress.
biotic stress. In this case, we don't know because it seems like Ramularia, but we do not know for sure. Okay. Because I do not have anyone in phytopathology in my lab. And this is something that could be potentially very interesting, is to align both of them and to look at that. Yes, there is definitely a differential response between waterlogged and biotic stress compared to control. Yes.
Fantastic. So from George Laurie, great talk, Sonia. My question is, did you take any of the trials to harvest and look at the quality of the grain?
This is a very good question as well. So the only thing that we looked was to protein content. We could clearly see from, we are still doing that for this year data, so I still don't have that data to tell you, so that's why I didn't present it. So our research assistant Tim is still drying the samples in the oven. We are talking about thousands of samples, and looking into the protein content from last year data. Our pilot study is that we could clearly see statistically significant reduction on protein content of waterlogged plots compared to the control ones. And we are also going to look into beta-glucans as well. So, but only this year, because this year was the big year. Last year was just a trial. Yeah.
Okay, great. So from Tom Mulholland at Chivas, do you expect to be able to use your techniques to determine optimal farming techniques? For example, optimized fertilizer applications and timings, even in waterlogged conditions?
Yeah, that's the whole purpose of remote sensing, is that you can estimate, and you can try to use this type of data to predict things, and that's why machine learning can be also very important. So our goal is to, not in terms of fertilizer applications, but I would say that in the future, yes, some of our things. So one of the end goals that we want is to, for instance, to predict which are the traits that you have to look at. Because when we are talking about this is a big data problem, I simplified a lot the story. So for instance, from the PSI instrument, I can easily have 200 traits, 200. So within this 200, which one is the ones that we need to look at. I cannot look at all the 200, you know? So that's when machine learning can really have an impact and say, this one, you can see a change on these days, and that one, you can see that change on that days. So using that knowledge, we can then build up for the future, yes.
Fantastic. So I see we've got a good friend from Israel on the line here, Isle Friedman. Great talk, Sonia, thanks. Concerning final grain yield, what would you predict as a favorable growth habit under waterlogging? For example, smaller or larger plants or developmental stage dependent changes? Nice to see you, Isle.
That's a very tough question. I'm not sure if I will be able to answer properly. One of the things that we thought that we are seeing, and we are trying, again, this is not part of this big project, because as I said, when we are looking into breeding or genetic studies, and in this case, it's a genetic study because we are having 230 lines, we cannot look into the roots. But we can look into the roots in a smaller scale. So this is what we are doing. We have small projects, like I would call them satellite projects in the lab, that we are trying to look at the roots. And one of the things that we observed, and it's very clear, is that this heritage barley has a much bigger and stronger root architecture. So this means that when you have the waterlogging impact, although there's a reduction, there's not such a larger reduction. So that could be something that I would say it's there is a reduction and you have a shallow roots, but it's still able to uptake the nutrients. The other thing that I would say it's very important is the nitrogen use efficiency. If you have a plant with higher nitrogen use efficiency, the plant will still be able to grow and use nitrogen more effectively than a plant that cannot. So I would say these two would be something to look at: roots, nitrogen use efficiency, and in terms of but the last thing that we want is really maintenance of yield. What we are doing when we are putting the waterlogging at this stage, at early seedling stage, we are affecting the tillering capacity. Okay, so the plants that are still able to produce tillers under this high pressure, environmental pressure, would be the one that in the end would be better able to cope with waterlogging. Now, if we would do, that would be again a different project. If we would do waterlogging at a later stage, more at the grain filling stage, then you would be affecting on the grain size. Okay, so it really depends on where you put the waterlogging and what type of physiological characteristics will be important.
Great. So we've still got a couple of questions here. I'm just going to go through them. Michael's getting a second bite at the cherry here. He says, do you also look into small or starchy granules or increased gel temperatures as a result of drought stress?
No, we are not looking into any of that. That again, it would be on the realm of very more focused research, and we are going into a larger number. So it could be something to follow up, but we are not doing that.
Okay. So Pete Headley asks, do you have any plans for conducting complementary studies such as transcriptomic analysis?
No, not having any plans. Again, in this project from Chigask and UCD before I came was the Wiki project. They did some transcriptomic studies. I'm not sure if they published already or not, but there is a little bit of transcriptomic studies being done. Now, I have to say that waterlogging has been extensively studied in terms of molecular analysis, and there's lots of very good people, really very good people looking into it. And the molecular basis, you go to ethylene responsive factors and the ND-GRON pathway, and several genes have been identified in strong candidate genes. So in terms of molecular responses, things are quite studied. I would not say totally studied, but have been well studied. While I felt that you always have to think about your research gap, right? And of course, you come to Ireland, you think about rain. So made sense waterlogging. You think about Ireland, you think about Guinness, Irish pubs, makes sense barley. And when you think about the gap in research is that you think that there's amazing research being done at molecular level, and then there's also lots of research being done at KTL level and double haploid populations and all of that. But I felt that in terms of physiology in the field, it was much harder to know, and in the field it's a totally different game ball compared to what you do in the glass house or in Arabidopsis. So I wanted to really see what happened in the field using a large collection. Yeah.
Thank you. I see time's getting on a little bit, so I'm going to ask one question, Sonia, and then I'll open the floor up just to see if anybody wants to ask a question out loud. They can unmute themselves and ask a question out loud. But my question, it kind of relates to waterlogging stress, but it's not exactly waterlogging, and that is, what's the impact of moisture on the crop canopy for all of the traits that you actually analyze? So either from dew on a nice cold morning towards the end of the season, or from rain the night before, even though it might be a fantastic day, the day you're actually going to fly your drone.
I really don't know how to answer that question, Robbie. I would have to go and look into it and discuss it. Because when we look, the thing is that when we fly the drones, you cannot fly the drones too early in the morning, okay? You usually only fly a little bit later after 11 a.m. or something like that. And this is because of the light. So you have you have to have the sun in a more upward position so that you can fly and really collect the data in a more meaningful way. So I would say that by the time you are flying, you would have removed all of that. You would not have the morning dew anymore, when you're looking into the imaging data. That would be my gut feeling, but I honestly do not know.
Okay. Yeah, it's a worry, I guess, for us as well, if we're thinking about using these types of field phenotyping methods, what impact some of the standard environmental conditions would have, like wind as well, for example.
Yeah.
Wind, moisture. Yeah.
Yeah, well, you have sort of all the weather conditions that you have to come up, right? So you can only fly under certain weather conditions. So if there's a little bit of rain, like, yeah, under either you're flying and there's a little bit of raindrops that fall down and that's fine, and it's not affecting. Or if it starts to have a little bit more of intensity, it's out. You have to immediately land. If the wind is too strong, you cannot fly as well. So it really depends on the type of again, the challenge is that it's very limiting sometimes. And in our case, it's the best way because we are doing in water logging. We have been discussing the use of just like in the ground, but then you could only do it after the water logging has finished, and otherwise if you have a muddy thing, so even getting the core of the soil, it was all smushy plashed, like a chocolate mousse, it's like totally mushy, so you cannot have wheels there. So and the wheels do not have all that impact. You can still collect data with high wind, so it's never an easy decision to
Yeah.
select the best system.
It never fails to amaze me the number of complications that you always find that you never expect when you start doing something, which is, yeah, I think it's the same for everything. So I'm going to open the floor up in case anybody out there just wants to ask a question by speaking. So if you have a question, please unmute your mic, stick your camera on, we'll see who you are, and fire away. I'll just for a couple of seconds. Sonia, nobody's chomping at the bit. So
No.
I should probably, is that somebody wanting to say something? I should probably say that in the chat box, there's lots of people have said, thanks for a great talk. And I think we would all probably agree with that. So thank you for a great talk. Really enjoyed it. I think you're doing some fantastic stuff across there. And I'm really glad to see that it seems to be going well as far as we can see anyway. So I think we've got one final slide to go up on the screen. Is that correct, Zoe? So just a little advert and a thank you. So the thank you will be for attending the seminar series we've had so far in 2021. We're in the process of trying to plan the next seminar series for 2022. So first of all, keep your eyes open and your ears open for announcements about future seminars. However, also we want to, if you have anybody that you would really like to hear giving a seminar, as part of the International Barley Hub seminar series for 2022, please either drop me or drop Donna or anybody else across here that knows anything about the IBH, drop us a line and say you'd really like to hear this person, and we'll get ahead and start sending out invites. So I imagine that we probably won't start now until February 2022. January seems like an awful cold month to start the seminar series, but I would just like to thank you again, Sonia. You've been a fitting finale to this series of talks for IBH, and it's been great to see you. And like I say, it's been great that you've had some attendees from all over the world actually, which is really nice.
Good. No, and thank you for thank you a lot for the invitation. It was really lovely to show what we have been doing, and a big thank you for my group because they do the hard work. They always do the hard work. Yeah, and if you have any questions or if you want to just chat with me, you have my email. You can always drop me an email. And yeah, Robbie and I will be in touch, and we have a couple of papers to publish, Robbie. So
you never know, we might actually be able to meet in person at some time.
Hopefully soon. Yes, yes, hopefully soon.
So thanks again, Sonia. Thank you everybody for turning up, and thank you for the commitment you've all shown as well to the IVH seminar series over the last 6 to 8 months. So look forward to the talks from next year again. So thanks again, Sonia. Thanks everybody for turning up.
Thank you.
Did I miss anything, Zoe?
No, you got everything, Robbie.
Oh, look at
Thank
that.
you.
An expert.
Bye. Thank
Sonia,
you.
take care. Lovely to
Bye.
see you.
Bye.
Thanks a lot
Thank you.
again.
Bye.
Bye-bye.
Yes, thank
Bye-bye,
you.
everybody.
Thank you.